Welcome to Human Physiology. Today, we are exploring the chemical factories of digestion, specifically the gastrointestinal associated glands. If you have ever wondered how the complex meals you eat actually turn into the microscopic energy your cells use, you are in the exact right place. Let us dive in and uncover this amazing system. To understand how these glands work, let us start with a simple analogy. Imagine you are running a massive factory assembly line designed to dismantle old cars. The conveyor belt itself is like your digestive tract, moving the cars along. But the belt alone cannot take the cars apart. You need specialized robotic arms sitting outside the belt to spray dismantling chemicals. In your body, these robotic arms are the gastrointestinal associated glands. They sit just outside the main tube and squirt essential juices to break down your food. Without them, the tract is just a dry, useless tube. Now that we have our factory analogy in mind, let us look at the formal biological definition. In physiological terms, these are specialized exocrine organs. This means they are anatomically distinct from the main digestive tube, but they are functionally crucial. Because they are exocrine glands, they produce specific secretions and release them through tiny tubes called ducts directly into the hollow space, or lumen, of the digestive tract. These secretions are packed with water, mucus, and highly specialized biological catalysts called enzymes that chemically cleave large food molecules into absorbable microscopic nutrients. Knowing what these glands do generally, let us identify exactly who these key players are in your body. The primary associated glands include the salivary glands, located in your mouth, the liver and gallbladder, situated in the upper right abdomen, and the pancreas, which hides just behind the stomach. We classify these as accessory digestive organs. Why? Because the food you eat never actually enters them. They sit on the sidelines. But as we just learned, their chemical contributions are absolutely mandatory for the digestive process to occur. Let us visualize exactly where these organs sit in relation to the digestive tract. Looking at this diagram, you can see the large liver at the top right, with the small green gallbladder tucked just beneath it. Notice the stomach is shown as an outline so we can clearly see the yellow pancreas sitting right behind it. The most important features here are the tiny tubes, the common bile duct from the liver and the pancreatic duct from the pancreas. See how they merge and empty directly into the duodenum, which is the start of the small intestine. This is exactly how those chemical factories deliver their products to the conveyor belt. Seeing their anatomy helps us appreciate their clinical importance, which is truly a matter of life and death. These glands are the absolute lifelines of human nutrition. For example, if your pancreas stops secreting its enzymes, a condition called exocrine pancreatic insufficiency, you could eat thousands of calories a day and still slowly starve. Your body simply cannot absorb unbroken down fats and proteins. Similarly, the liver produces bile, which acts exactly like dish soap to dissolve dietary fats. Without bile, fat-soluble vitamins like A, D, E, and K would pass right through you, leading to severe malnutrition. To understand how these vital glands function dynamically, we need to measure their activity mathematically. Physiologists use the secretion rate formula. Conceptually, this just tells us the speed at which a gland works. The formula is S equals V divided by T. Here, S represents the secretion rate, typically measured in milliliters per minute. V stands for the total volume of the digestive juice produced in milliliters, and T represents the time duration over which the secretion occurs, measured in minutes. If the volume produced increases while the time stays the same, the secretion rate goes up. Conversely, a damaged gland producing less volume over the same time will have a decreased secretion rate. Now that we have our formula, let us apply it to calculate a student's basal saliva production. We're given that during 14 hours of resting time, their glands produce 420 milliliters of saliva. We need to find the basal secretion rate. First, we convert the 14 hours into minutes by multiplying by 60, giving us 840 minutes. This standardization is crucial for medical metrics. Next, we substitute the volume of 420 and the time of 840 into our formula. Dividing these tells us exactly how much fluid is produced in a single minute. The final answer is 0.5 milliliters per minute, which is a perfectly normal, healthy trickle of saliva. Building on that basal rate, let us see what happens when the student actually eats. We are given that during 2 hours of active eating, the glands produce 360 milliliters of saliva. Just like before, we convert the 2 hours into minutes by multiplying by 60, which gives us 120 minutes. We then divide the stimulated volume of 360 by the 120 minutes. The final answer is exactly 3.0 milliliters per minute. Notice how this active rate is a massive 6 times higher than the resting rate. This dramatic increase shows how powerfully the brain activates these glands the moment you taste a delicious meal. Finally, let us combine these two states to understand the overall daily workload of the salivary glands. We simply take the resting volume of 420 milliliters and add it to the stimulated volume of 360 milliliters. This simple addition gives us the big picture. The final answer is a grand total of 780 milliliters of saliva produced per day. Physically, this means nearly a full liter of fluid is pulled from the blood, turned into digestive juice, and recycled back into the body every single day. It is a beautiful demonstration of how hard these associated glands work. Beyond just the volume of fluid, we also need to measure how potent these juices are, which brings us to our second formula. This is the standard concentration formula used in chemistry and physiology. Conceptually, it tells us how densely packed the enzyme molecules are within the fluid. The formula is C equals M divided by V. Here, C represents the concentration of the enzyme. The symbol M represents the mass of the enzyme secreted, measured in milligrams. The symbol V represents the total volume of the pancreatic juice, acting as the fluid carrier, measured in milliliters. A high mass in a small volume means a very potent, highly concentrated digestive juice. Let us put this concentration formula to work with a clinical example involving the pancreas. Suppose a patient's pancreas secretes 1,200 milligrams of the enzyme amylase into a total juice volume of 800 milliliters. We need to calculate the concentration to ensure the juice is strong enough to digest carbohydrates. We take the given mass of 1,200 and place it in the numerator. We take the given volume of 800 and place it in the denominator. Dividing 1,200 by 800 gives us our final answer of 1.5 milligrams per milliliter. Physically, this means every single milliliter of juice contains exactly 1.5 milligrams of pure carbohydrate-destroying enzyme, which is highly effective for normal digestion. We have covered a lot of ground today, So let us review the most important ideas. First, remember that the gastrointestinal-associated glands are the unsung heroes of digestion. While the stomach provides mechanical mixing, glands like the liver and pancreas